topic dissolved oxygen, BOD and COD. And these are the general parameters of water pollution. Generally, the different parameters that determine the nature and the extent of water pollution include both physical, chemical and biological parameters. The physical parameters include the color, odor, turbidity, density, temperature, etc. of the water. That is, if you observe the water and noted its color, odor, turbidity, density, temperature, we can find out or we can determine how polluted that water is. And also the chemical parameters, that is by measuring the chemical parameters like pH, total dissolved solids, that is TDS, and their ionic compositions, and by measuring the suspended solids, and also by measuring the dissolved oxygen, residual chlorine, and chemical oxygen demand and biological oxygen demand, redox potential, and by chemically determining the radioactive substances, organic materials, metallic ions, oxides, byproduct of industries, and so on, we can find out the pollution of the extent of pollution of water in a given sample. The different biological parameters include different types of microorganisms, bacteria, algae, small animals such as protozoa and crustaceans, and so on. And these are the general parameters used in the determination of the extent of determination of polluted water. In our syllabus, we have to study the dissolved oxygen, biological oxygen demand BOD and COD. Let us discuss in detail about these topics, that is dissolved oxygen, biological oxygen demand and chemical oxygen demand. First is dissolved oxygen. It is simply designated as DO. The dissolved oxygen content in water is an essential requirement for aquatic life. The normal level of dissolved oxygen in a water body should be 4 to 6 ppm. Decrease in this value indicates that the water is polluted, and this pollution is mainly caused by organic matter, for example sewage, industrial waste and runoff from agricultural lands. The large proportion of bacteria decomposes organic matter present in water. They consume the oxygen dissolved in water. The amount of oxygen that water can hold in a solution is limited. In cold water, the resolved oxygen can reach a concentration up to 10 ppm. That is, the maximum concentration of dissolved oxygen in cold water is about 10 ppm, whereas the oxygen in air is about 2 lakhs ppm. That is why even a moderate amount of organic matter, when decomposes in water, can deplete the water of its dissolved oxygen. That is, oxygen reaches the water through either atmosphere or from the process of photosynthesis carried out by many aquatic green plants during daylight. However, during the night, photosynthesis stops, but the plants continues to respire, resulting in the reduction of dissolved oxygen. The dissolved oxygen is also used by microorganisms to oxidize organic matter. There are several approaches to evaluate the dissolved oxygen content of water samples. In chemical methods, water samples are treated with manganese sulfate and alkaline potassium iodide solutions containing sodium acid. The dissolved oxygen then reacts to produce an equivalent amount of free iodine. After acidification of this solution of iodine, and then it is treated with the standard sodium thiosulfate solution, we can find out the amount of dissolved oxygen in that sample of water. This procedure is subject to a number of interference effects, and also great care should be taken to avoid the trapping of atmospheric oxygen during the course of titrations, particularly in the reaction vessels. The general routine method for monitoring dissolved oxygen is using the electrical cells. These are being widely used. The electrical cell unit basically consists of two solid metal electrodes in contact with a fixed volume of electrolyte solution. By applying a suitable potential, the dissolved oxygen gets reduced. The magnitude of the resultant current flow can be calibrated in terms of the oxygen concentration. Hence, by using this method, we have to find out the dissolved oxygen in a given sample of water. Next is biochemical oxygen demand. It is also called biological oxygen demand, that is BOD. It is the amount of dissolved oxygen needed by aerobic biological organisms to break down organic materials present in a given water sample at a certain temperature or a specific time period. Or we can define the biological oxygen demand as the amount of dissolved oxygen used by microorganisms in a biological process of metabolizing the organic matter present in water. The more organic matter is present in the water, the greater the biological oxygen demand, and the greater the value of BOD. The lower the amount of the dissolved oxygen available for higher animals such as fishes, the BOD is therefore a reliable gauge of organic pollution of a body of water. One of the main reasons for treating wastewater prior to its discharge into the water resource is to lower its BOD, that is, reduce its need of oxygen and thereby lessen its demand for the stream, lakes, rivers, and estuaries into which it is released. If there is too much organic matter is added to water, all the available oxygen is used up. This causes oxygen dependent aquatic life to die. Thus, anaerobic bacteria which does not require oxygen is begin to break down the organic waste and produce chemicals that have foul smell and are harmful to human health. Aerobic, that is oxygen requiring bacteria, degrade these organic waste and keep the water depleted in dissolved oxygen. Thus, the amount of oxygen required by bacteria to break down the organic matter present in a certain volume of sample of water is called biological oxygen demand. The amount of biological oxygen demand in water is a measure of the amount of organic material in water in terms of how much oxygen will be required to break it down biologically. Clean water would have a biological oxygen demand value of less than 5 ppm, whereas highly polluted water could have a BOD value of 70 ppm or more. Next, we have to discuss the chemical oxygen demand. The chemical oxygen demand is a test which is used to represent the amount of chemically digestible organics present in the polluted water molecule. COD measures all organics that are biochemically digestible as well as all the organics that can be digested by heat and sulfuric acid. It is used in the same application as that of BOD. COD has the advantage over the BOD that in the analysis can be completed within few hours, whereas BOD analysis requires more than 5 days. That is, around 5 days we get the result. The major drawback of COD test is the presence of hazardous chemicals and toxic waste disposal. The COD theory is like the BOD test. Here, the oxygen is used to oxidize the organics to carbon dioxide and water. However, instead of free dissolved oxygen, chemically bound oxygen in potassium dichromate is used to oxidize the organics. That is, in COD, chemical oxygen demand, here using the oxidizing agent potassium dichromate. As the potassium dichromate is used up, the Cr3 plus ion is produced. The amount of dichromate used is proportional to the amount of organics present in that polluted water, that is water sample. Likewise, the amount of Cr3 plus ion present is proportional to the amount of organics gets digested. That is, this COD process using potassium dichromate involves two steps: digestion of the sample with dichromate in acid media, that is the organic matter. It is represented by this carbohydrate. It is represented by Ca, Hb and Oc, where A, B and C are numerical numbers, plus potassium dichromate plus H plus, that is acid, gives carbon dioxide plus Cr3 plus and water molecule. The titration of the excess dichromate with Fe2 plus, that is, it is converted to ferrous ammonium sulfate after the carbon oxidation in the organic matter, that is Cr2O7 2 minus plus 6 Fe2 plus 14H plus gives Cr3 plus Fe3 plus 7 H2O. That is, the organic matter is converted to water. This method uses test tubes with pre-measured amount of potassium dichromate, sulfuric acid and catalyst. For the digestion to occur, the reaction needs acid, heat and a catalyst. The acid is commonly used as the sulfuric acid and is already in the test tube. The sample will get heat when the sample is added to the acid once it is mixed. Be sure that the cap of the titration vessel should be tight before mixing and just prior to placing into the digestion reactor. References: Environmental Chemistry Fundamentals and Environmental Chemistry Unit 14 NCERT online text.